welcome to this talk, and I will ask Professor Krishna to introduce Sarit. He's the host of this lab visits at biochemistry at BHU. So, Professor Krishna, can you please,
Okay, thanks, Dr. Padda, for introducing me. Hi, Sarit, how are you?
Good. Nice to meet you.
Glad to see you, and welcome to the Department of Biochemistry. We have, I think, roughly about 12 to 13 participants, okay, ranging from UG to PG as an audience, along with our research students here. And they're all waiting for your exciting talk, okay. Before we formally move, I just would like to introduce to all of this audience about Dr. Sarit. Dr. Sarit Agasti is an associate professor currently at JNCSIR, Bangalore. He received his PhD in chemistry from University of Massachusetts, Amherst, where he worked under the guidance of Professor Vincent Rotello. He subsequently joined Harvard University to pursue his postdoctoral research, where he worked with Professor Ralph Wiesler and Professor Pen Yin. Dr. Agasti research group focuses on developing new chemicals or chemical tools for biosensing and bioimaging especially. He has been selected as Young Associate of Indian Academy of Sciences, Bangalore. He is also the recipient of prestigious DBT Wellcome Trust India Alliance Intermediate Fellowship, the Innovative Young Biotechnology Award, Fellow Merck Young Scientist Award, and the INSA Medal for Young Scientists. Just briefly, I introduced about Dr. Agasti. He has much more credentials and he's very young and dynamic researcher, positioned at chemistry department, if I believe. Okay, and he's expert in molecular tagging, for imaging purposes beyond conventional microscopy. Today he'll be talking about molecular tags for imaging beyond traditional microscopy. I'm sure you will enjoy the talk. Okay. Yes, Dr. Sarit, please go ahead. Thank you.
Sure. Thank you. Thank you for the invitation. Today, what I'm going to discuss with you today about the flavors of microscopy and what goes behind looking at molecules in the cellular system. And so, what you see here in this slide, it's a very closer look to one of the devastating form of cancer, breast cancer cells. And here you can clearly see that the molecules are light up in different colors. You can see a kind of yellowish color and a bluish color all around. So this yellow color, what you see here is essentially is one of the important molecular marker, which is the HER2, which is overexpressed in this sort of cells, and which tells us that these cells are essentially the cancers. So this tells you that you are able to visualize a specific molecule which are really interested in the health of human beings, and will be able to investigate into its intrinsic details. You can clearly see the distributions of these molecules. So it's also important to the overall health of the human being that you really need to look molecules into different contexts. And then when it comes to the basic science, what I show you here, it's a bit different molecule. It's an actin, which is a very important component for almost every parts of every process of cells. So this molecule is what you see here is organized in an entirely different manner, which you saw it's in the previous slide. Right? It's a distribution which looks amazing, and it has a distribution which is present in the tissue of the dorsal. Right. So again, you see that this is a tissue which is a muscle tissue, and the distributions of molecules here are entirely different than what you have seen before. So you can clearly see that when you want to look at the molecules, you not only would like to see whether they are there or not, you'd also like to understand that how they are distributed within the tissues, right? So that governs the function of this model. And let's go even more macroscopic scale, and you go down to the scale where all the process originates, which is the cell, right? And now you see that we started from much bigger samples like in tissue, and then we are coming down towards much smaller scale samples like for example the cell. And now if you go inside the cells and try to really understand that which are the molecules which are very important for the function of the cells, one molecule comes really into mind is something like a microtubules, and those are one of the most important cytoskeleton protein which is present in the cell. And you can clearly see that these are basically the fibrous the filamentous structures which are present in the cells. And also you can see that this is what essentially you would be looking at, but let me show you what is the exact structure of this bond. So you can clearly see that what we see by traditionally looking at under the microscope, and what are the really original structures. And you can clearly see that they're looking quite different and looks much more clear and much more crisp. And you can clearly see what their distribution looks. And the important point, what I wanted to mention to you here, that the first image what I showed you, the tissue sample is essentially a millimeter scale or even a centimeter scale size. And then the muscle sample was close to a millimeter samples. And what I show you here is a sample which is 20 nanometer in diameter. Right? So you can clearly see the distributions or the length scale where the biological phenomena happens, right? So it starts from nanometer scales, 2 millimeter scales to centimeter scales. And what you are really seeing here, it's one of the important tools which are able to probe the phenomena or the structures and function of the molecules, starting from the nanometer scale all the way to the millimeter scales to the centimeter scales, is the fluorescence language. And that's probably one of the important tools, I would say, that which are able to do that. So now let me tell you that how these images are acted. Just for the beginners, this could be a quite important process for you to know that in order to understand from so this is my pointer is visible. Yes.
Yes, sir, it's visible.
So now in order to go from pretty much a boring cells, where you don't really see them in any colors, to a really beautiful image like this, you need two important or two major components. So first major component you need to give these cells some specific colors, right? So that you can specifically tag this molecule. Then that's what it comes as the molecular tags, which you see here is number one. And many times the most widely used molecular tags here is essentially antibodies, which are usually labeled or tagged with something like a fluorescent molecules. And the second important part, which is also plays a critical role in acquiring many of these images, are essentially an optical microscope, because that is something what you really need to use in order to translate the things which you are in the cells into a beautiful image like this, right? And these two things usually plays the crucial roles and works hand in hand to come up with this image. And there are development in both of these fields which is happening all around. And I would say that the first part is usually dominated by the principles of chemistry, because these are associated with molecules and the development of probes. And second part usually dominated by the principles of physics. And many times also you really need to process these images. So some sort of analysis also associated with all of these phenomena. And being a chemist, I am going to majorly focus on this part where the knowledge of chemistry, chemical tools, or molecules can be developed, which can be now combined with the advancement of the microscopy to reach into a phase where I showed you that it will be possible to look at from disease samples, which are centimeter scales, and all the way to nanometer scales spanning across different lengths. So now going towards talking about the development of the probes and how it related to microscopy, I would like to first tell you that why we are interested in fluorescence. I'm sure there are many other modes of imaging which is coming across. For example, mass spectrometry based imaging. There are many times Raman based imaging, and there are various other types of imaging modes are also available. Even nowadays, people are interested in looking at transmission electron microscopy or scanning electron microscopy. But the fluorescence is something which, as I told you before, that it can give you across the length scale, right, from centimeter to millimeter. In addition to this, you also get a benefit of high contrast imaging because your signal is against the dark background. And very importantly, they are highly specific. Like if you are interested, if you're a molecular biologist, you want to look at a specific molecule, you can color them with specific tags. For example, you can use an antibody, or you can use an expression of proteins like GFP, and also using small molecules to give them specificity. In addition to that, you also have multicolor imaging. That means you are not limited by the dark and white background, which are usually seen in TEM images also. Here, you can essentially look many different molecules by providing them with different colors by using different fluorescent probes, and then you can look at the multicolor rates. And very importantly, one of the most important advantages that you can actually look at them while these samples are alive. So that is called the live cell imaging, and I think that is one of the most attractive features of fluorescence microscopy, because it's an optical imaging. So you essentially have the benefit of very gentle radiations, which are not harmful for the cells, and as well as your detection modes and everything are highly compatible to live cells. And even you can maintain the cells in a nice environment inside a microscope, and you can look at them while they grow, they distribute, and they actually do function. And many times you can reach into a quantitative estimations of the molecules. Right? You can quantitate how many molecules are present, and you can use them for analysis. And also, you can develop probes which can act as the sensors. For example, calcium ion sensor, pH sensor, which has been wide. So this has been quite advantageous to use fluorescence for many of these. So that's why not look at what is fundamental principle behind the fluorescence. Right. So the important thing what you look at fluorescence is essentially you excite a molecule, right? When you use a specific wavelength of light, and then these will be emitting a wavelength of light, which is usually the longer than the wavelength of excitation light, and that's why it's called that you are detecting the molecules in the dark backgrounds also. And also, cells does not have most of the times this sort of an fluorescent molecule present in this, right? So that essentially gives you a highly contrast image. So you excite a molecule and you collect the light which comes out of it as a fluorescence. And then let's look at what kind of molecules we really need to use. And this gives you an idea that what essentially this fluorophore looks like, and what are the properties of the fluorophores which are essential for biological imaging parts. So here you can see that the molecules are distributed in the wavelength starting from UV all the way to IR region. And then as you go close towards the IR region or near IR region above 600 nanometers or so, you essentially started to look at molecules, much deeper samples without much much backgrounds, and that gives you much more abilities to look at. In addition to increasing the wavelength of the fluorescence, fluorophores, other parameters which you really look at something called the brightness of the. So brightness means that how intense your signal is going. Right. For example, if you want to look at highly sensitive phenomena, like if you have a very low number of molecules present in your cell, you really would like to get them high intensity emission coming out. And that brightness is defined by two parameters. One is how is the epsilon coefficient, like here you can see the epsilon coefficient, and also the quantum yield of this molecule. So this absorption coefficient is something which tells you that how many number of photons you are going to absorb, and the quantum yield going to tell you how many numbers of photons you are going to emit after that. So both of these parameters is essentially the one which tells you that what is the brightness of this molecule. So based on that, these has been given, and as you can clearly see, some of the very well performing molecules over here, like cyanine dyes, the rhodamine dyes, OTP dyes, are the ones which essentially constitute the molecular scaffold, which are really is essential to do those sort of things. Right. So these are the areas what we are trying to look. Where you can see is majorly populated by these sort of cyanine dyes, and many of you may have heard Alexa dyes, right, which are very common and majorly people are used, and many times people use Atto dyes, right. So those are all falls into this sort of dye. Right? So those are majorly based on xanthine dyes, which are these are all the samples. So these are a number of years of development where we are actually able to make. We were able to make different sort of fluorophores with advanced property, and that has immensely contributed for going from the fluorescence spectra, right? Actually looking at molecules how they fluoresce to the fluorescence image, right? So you use this molecule where well-known demonstrations that how these molecules can be used. In addition to that, you can also essentially use molecules like green fluorescence proteins. Which are even has been awarded to the Nobel Prize as well. So those are the ways you can essentially go from a fluorescence spectra to look at fluorescence imaging. So what is the difference between fluorescence spectra and fluorescence imaging? Is that in fluorescence imaging, you essentially integrate the spatial information, right? So not only you record the fluorescence emission coming out of it, but you exactly know from where the fluorescence spectra is or fluorescence emission is coming. So you have the coordinates, you have the spatial information integrated, and that's what it gets much more interesting. And this is one of the very simple fluorescence imaging setup what you can think about, which constitute as I talked about to you, that you prepare a sample which contains of fluorescence levels. And in addition to the fluorescence levels, you need a microscope, right, which should be able to record this image, and which contains, as you can clearly see here, that you need an excitation source, and most of the time it's going to be a laser, and you need in a microscopes which will be able to integrate these lasers, and able to have a really highly sensitive camera which will be able to record and give you the information. So now going beyond the basic principles of having a fluorophores and having a microscope, let's look at what are the desirable in terms of fluorescence microscope, right? And we kind of try to combine or try to work on these four different areas and try to see how we can develop or advance this field. So what are the four different criteria or important parameters which is essentially needs to be the challenging parameters to be developed or it has to be improved, that concept that where you really need to go for resolutions where you should be able to look at the molecules in its molecular resolution, right? For example, if you think about it in a protein, this will be around 2 to 3 nanometer of size. So you really need to go down a nanoscopic resolutions, which should be below 10 nanometer or so. Then another important parameters which you can see is clearly needs development is the multiplex, right? So multicolor or multiplex imaging can be done by using fluorescence. However, as you know that fluorescence spectra are quite broad, right? And because of that, you don't have abilities to resolve beyond 4 different colors also, right? So in a microscope, it's quite limited. You can only use a certain number of fluorophores to get fluorescence spectra, right? So that is limited by the fluoro, right? So you have only 4 different colors you can resolve. So this is quite limited. And the third important thing, absolute biomolecular quantity, right? So you would like to quantify the number of molecules, not only relative quantification, but you really want to know exact number of molecules. Is it going to be 5? Is it going to be 10? Not like a relative to some. Then another important parameters which we constantly look at, that how do we able to do long-term dynamics of individual bio, right? So if you are interested in looking at life cell, many processes happens in hours, right? Even days. So can I have the abilities to understand or to visualize molecules for really, really long? So whatever I have shown you in the development, so there many of these parameters are just not satisfied utilizing the standard strategies what is able to provide by using fluorophores or fluorescence microscope. So our group essentially works on a few of these areas and then try to develop molecules so that we are able to solve such challenges in collaborations with many of the in-house collaborators as well as collaborators in nearby institute like ISC. So I really thank them for thank my group for providing such a support so that we will be able to advance some of these areas. So now coming to the point that we have shown you that you have a microscope and you have a fluorophore, right? So you can essentially couple a target, right, for fluorophore, and you can go to a microscope and record the image. And that's the most simplistic version of looking at the microscope. However, you can think about that not only you can attach these two parts, like target and your label or the tags, through an interactions which is stable and non-breakable. What you can say a covalent bond. Like, there are two types of bonding: covalent and non-covalent bonding. And the covalent bondings is the one which are difficult to break. So you essentially most of the time you attach them to a covalent bond. But what I'm going to tell you today is that what happens if you try to attach them to a non-covalent. And as you know, the non-covalent interactions are dynamic in nature, right? So they are reversible and they can make and they can break and they exist in the dynamic. And what I'm going to talk to you today, that how these sort of molecular tags are essentially going to be useful for something where we are able to solve some of the challenges what I just discussed. Oh, let me introduce you with an essential molecular tags or the non-covalent tags. What we essentially use in this sort of an aspect is called this sort of a macrocycle. And as you can see that these are kind of a cavitant molecule. So you can see that this has a cavity. Not going to in details, but these are the molecules which are essentially have essentially no toxicity onto the cells. And these are high solubility, non-toxic, many times cell permeable in nature, and something called bioorthogonal in nature. So bioorthogonal means that this molecule does not interact with any other molecules in the cell. So that's what I say is that these are orthogonal to biological molecule, and they don't have. So this molecule also does not present in the cell, right? So these are essentially a synthetic molecule which we create in the lab. Now, what is the feature of this molecule? So this molecule has the abilities to interact with, so let me go back to this slide first. So this molecule has the abilities to interact with certain other molecules, right, which are again not present in us. So essentially you see that these are range of the molecules, right, again synthetic in nature, and these are some of the amino acids also it's possible. but special types of amino acid, I must say. So these are some of the category of molecule which this cavitant molecule is able to interact. So these are able to form some sort of non-covalent interaction. So these hydrophobic part of the molecule essentially can go inside this cavity, and this cationic part essentially interacts with the rim of the molecule or the portal of the molecule. So that's how they make the kind of host and guest compound, and these are usually reversible. And these are the complex which you can essentially modulate in terms of their binding affinity, right? So the molecules what you choose as a guest or the essentially interacting with this sort of a cavitant molecule can have different types of binding affinity, starting from you can choose a binding affinity which is very high and can compete or even overpowers something which is available in the biological systems like the biotin and streptavidin. Right? Biotin and streptavidin, I think, is used almost all biological laboratory for many purposes. And these are the molecules which is a small molecular component and can beat the affinity which is present in the biological system and achieve an affinity which is 10 to the power 10 or 15 range. But what are the other advantages of these using molecules is that its abilities to tune down its affinity, and then you can reach somewhere like where you have a low affinity complex, something like which is called reversible. In addition to this, you can also achieve where the molecular complexes can form in the presence of a tree, right? So these are the three types of modularity which gives, because you have a tag which is non-covalent in nature, right? As you can understand, if it is a covalent tag in nature, you will essentially have the first part where you are able to make a bond between your two molecules, which is useful for many imaging, but you are not going to get this sort of a molecular tuning abilities which are present in this. So I will show you in this few slides that how this sort of a molecular engineering can do wonders in terms of looking at the biological system in terms of imaging capability. Okay, so let me first tell you that the first question we asked to this system is that are these sort of a molecular complex are stable in the biological system, and are they going to be useful for biological applications, right? So first thing what we did is that we took in vitro systems, which here you can see is essentially a DNA origami. So these are the kind of structures which you can make in the laboratory, and this has a defined size, and you can also program to place the molecular components in this system so that they have a defined distance and defined arrangement. So this here you can see that we took an 60 by 90 nanometer size DNA origami structures, and we placed these molecular complexes. You can clearly see here the same host and guest molecule onto these four sites. And then after placing them four sites, we first took an AFM image, which tells you that these structures are essentially formed, right? So these are the AFM image, and as I told, there's a 90 by 60 nanometer rectangular shapes origami, and those are present here that you can clearly see. But now I'm going to show you that are these molecules are having this complex in it or not. So this is what it looks like when you recorded this molecule under the microscope. And as I told you, we placed this sort of in four different forms. And when you looked at under the microscope, if I zoom in, you will clearly see that each of these molecules contains these four different atoms, right? So what it proves that yes, under an in vitro conditions, these are the molecular complexes which are able to form specifically and able to be stable in this context, right? So next, what we did is that we integrated these host-case pairs in some biological context. So what we did that we took these molecules into the cells, and we attached them into an important component, as I discussed, in the microtube. So essentially, your microtube will now have this host-case complex placed into this, and you are looking at the images after placing this post-response. And one of these molecules, what we have made is this, cuckold between CD7, Alexa 647. Essentially, these are used as a molecular tag to see whether the presence of this microtubule and whether they are specifically linked. And as you can clearly see in this image, that by utilizing these non-covalent interactions, and again, let me tell you, these are high affinity interactions, right? So which are close to something what you will be getting in biotin and streptol. So now this is a new sort of a molecular tags which are able to do this specific molecular tagging in cells. And also, not only you are able to see it using something like an antibody as you described before, you can also attach this molecule into a small molecule. Like over here, you can see that we developed a probe which is targeted to the actin. So this is a well-known binder called phalloidin. And now what we do, we label the phalloidin to our molecular complex, and then we put them in the cells, right? So these are the molecular targeting mechanisms which can also be used like both antibody as well as small molecule, and these are both compatible to this sort of an imaging. Now, not only in terms of using it in the cell, as I told that you can train these molecules to be highly specific in tissue. So this is an image which is acquired in our lab. And then this shows that how the distributions of actin in Drosophila ovary tissue, right? And as you can clearly, I have shown you before that how the distributions matters and how the distribution of molecules are different. And as you have seen before that how distributed in muscles, and as you can see here, distributions in ovaries. And you can clearly see they are quite different and very interesting structure. And this is what I showed you is essentially in the muscle. So you can clearly see that actin's distribution in this sort of different tissues are quite different, and those are able to be probed by utilizing this sort of molecular complexes which are entirely synthetic units. So these were the first of the examples. And as I told that dynamics, how do you probe the dynamics? So that you need to make molecules which will be able to level the cells in live cells and able to show the dynamics, right? So here what we did is that we made a molecules with conjugation to Taxol. So Taxol is something which is a well-known binders of the molecule, right? And then we put these molecules in the cells, and as you can clearly see here, these nicely level the cells. Not only level the cells, now you can clearly see that these are the mobiles, the dynamics, right? So that tells you that these molecules are not only able to level and image different molecules in the cells and tissue, but also able to probe something which is much more dynamic, right? So whole range of molecular complexes we are able to see. And also you can see here that you can actually look at these sort of things for really long time. And this is an image which was taken over a day. And as you can clearly see that how the cells are moving, how the cells are changing their shapes, and you can all do this by utilizing this sort of experiment, right? Now let me show you other images. So these are also distributions of molecules in the brains, and then these are the things which are present in the intestine. So these are the distributions of microtubules in intestine tissue. So these are something called the cuticles. These are the kind of cuticles present in the Drosophila, and all these can be imaged by utilizing this. So now you have a clear idea that how we combine fluorescence to the labeling to the image, right? So there is a combination of things. So you started a development of fluorophore, then you start a development and how these fluorophores are attaching to the molecules, right? And then you finally have an imaging protocol or the microscope which are able to take them. So you are able to see how this area works. Now let me tell you that so far I showed you that these are the ways the standard microscopy works, right? So now let's say we would like to see that the challenges. Can we go beyond the traditional fluorescence microscopy? what is meaning by going beyond, right? So if you think about traditional microscopy, it's highly limited. One of the biggest disadvantages of the molecules is the resolution. What do you mean by resolution? Is the something where you are able to resolve the actual structure of the bond. And if you are talking about the optical microscopy, they are limited by the Abbe's diffraction limit, which essentially tells you that the best resolution what you will be able to get is something by lambda by 2, close to it, and anything beyond that you would not be able to do. So if you think about that, if you have structures which are around 100 nanometer or 200 nanometer, which will be impossible for anyone to do. Right? So here you can see that if 2 points or if your distribution of microtubules are above than let's say 200 or 300 nanometer, then only you are able to separate them. However, if it is not above that, you will not be able to, if you are not able that, you will not be able to reach. And this is a fundamental limits of light. And that's why we really need to go beyond the diffraction. And how do we think about breaking this diffraction limit by new imaging approach? Right. So this is one of the area which we work on, and there are methods available, and many different labs works on many of these areas, looking at how to do this. I'm going to tell you specifically how we achieve these sort of strategies where we can go beyond, and this is entirely based on that how do we attach the probes or attach the molecules to your target, right? Based on how the chemistry works. So before going that, how to achieve this strategy, I would like to tell you that, what is the biggest challenge, that why we are not able to resolve. And as I told you that, let's say, this is a normal distributions of different targets. So each target is labeled with the fluorophores, right? So these are all the fluorophores, and here I am showing the fluorophores in its natural or in its inherent length. So a fluorophore will be a nanometer of size. So each of this by 1 nanometer. However, when you are detecting this fluorescence from each molecules, they will have a diffraction limited spot, as we call Abbe's diffraction limit, and you will see them in each molecules when you are detecting will have such a distributions, right? And this is also called the point spread function of the molecules, right? So this will be, and then all the molecules are essentially fluorescent at a time, right? So each the image what you will be getting in your microscope is essentially will be looking like, right? When each of the molecules are looking for, right? So you don't see the original structure. However, let's think about the situation that if we think about a strategy where something called a fluorescence blinking on and off combined with the single molecule localization, what does it mean? That you essentially have something called a single molecule localization. So if you have abilities to look at a single molecule, and you should be able to fit it with a Gaussian fitting, you would be able to get the nanometer precision localization from this single molecule. However, the problem happens when you have multiple molecules, right? So the Gaussians of each will be overlapping and will not be able to resolve. What about we, think of a strategy where instead of all the molecules being in a fluorescence on state, only few molecules are in a fluorescent on state, right? So there is no overlap between neighboring molecules. So essentially you have a fluorescence on, and there is no other molecule nearby. So you can easily feed them with a Gaussian and get a nanometer precision distribution, and then you turn it off this molecule and then turn on another molecule so that you can precisely localize, right? So essentially instead of having all of them in the on state, you make them on and off, called fluorescence blinking, and then you apply this single molecule localization. You finally come up with this strategy where you essentially have all the localization possible is aggregated and you get the structure, right? So this is what you can see that you have a problem where all the molecules in the fluorescence on state, and that it does not allow you to resolve the molecules in its original state. So you turn on and off the molecules and call fluorescence blinking, and you come back to the strategy where you can get this molecule into its original dimension. So now this has been well practiced in many different labs, has been working towards this sort of a strategy. And what I will tell you here is that how we are able to do, right? So there are mechanism present where you can take a fluorophore and we make a buffer, and you actually able to do a fluorescence on off cycle. And what I will tell you will different strategies. And I'll tell you that couple of advantages why we develop this strategy towards this. So here it's called that blinking by dynamic molecular interaction. So as I told you before that molecules are associated together either by covalent or non-covalent. So here we are talking about non-covalent bond, means the interactions is dynamic in nature. So which is essentially this middle part of it. Okay. And what you can see here, if you are in the middle part of it, and we are able to observe this molecule over the time, because of the dynamic nature of the molecules, you will be seeing them in an associated state, right? Sorry, first start with an dissociated state, so you don't have a fluorescent signal, but when there gets an associated state, you should be able to see a signal. And as they are dynamic in nature, over the time you will see them the dissociated state, and then again associated state, and this will go. So you see there that essentially in this case, I have a molecular complex which provides you with the blinking nature, right? On and off signal. So this cartoon demonstration will show you this in more details. Essentially, this is my target. So I label this target with my molecular complex. And what happens? My fluorophore binds here, but not with all the target. So that's how the distribution works, right? So you essentially have molecule target. So you localize them, and then it binds to another target, dissociate, target, dissociate. Right? As you can clearly see here, your fluorophores are not attached permanently, but they are interacting dynamically, and they are able to give you the necessary fluorescence. And based on that, two different targets we have designed. One is based on the DNA-DNA interactions, which is called DNA paint, and the other strategy which I showed you here is based on the use of this macrocyclic based system, which I just described a few slides ago. DNA-DNA interaction, why we use? Because there are two advantages. You can really modulate the binding strength between two DNA by just changing the base. So that gives you an idea that what would be the time frame that your molecules are in on and off. And the second important thing is that you can use this DNA and you can do a multiplexed image. Means if you just change the DNA sequences, you will be able to target a different molecule, right? So that essentially gives you an idea that you can probe infinite number of molecules, because there is a number of orthogonal DNA sequences you can generate around, right? So here you are not limited by the fluorophore, right? So that's the biggest advantage what you can get if you just tune the molecular tags not by covalent but non-covalent, and use a tag which has abilities to do multiplex. Not the fluorophore, but the tag essentially governs your multiplex. Okay, so I will show you how this strategy works, and this both applies to both DNA-based systems as well as this dynamic host-case systems. So what we did is that we tagged these molecules into the cells by using antibody. So we prepare something called DNA-labeled antibody, right? And then we tagged the different parts of the cells with the DNA tag, and we did this blinking. So how this blinking looks? Then this is an example, and you clearly see that really crisp, clear single molecule spots appears on your microscope, right? And see here, our biggest advantage is that here I am not doing any complexity in terms of my microscopic activities. I'm not changing in a laser. I am not using any other buffer. I'm not using any specialized fluorophore. Any fluorophore can be used, right? I just am utilizing the interaction, how this works, the interactions between two molecules. So this goes on blinking, and then you localize them in a Gaussian. So you fit it in a Gaussian, localize them, and that's what you see. So you see this. This is under confocal image, and this is what we get when we actually tune to this sort of multiplex super resolution. So right, you can clearly see that it is much better than your standard diffraction limited image, right? So the first image is essentially acquired by one of the best system. What is widely used is the confocal microscope. So the confocal microscope image and the real image, which is essentially the real characteristic of the molecule, you can see clearly. Let me show you in some quantitative manner. So here you see in the super resolved image, the microtubules now you clearly see is not at all a single line, what we are used to see, right? You can clearly see that they are kind of a two lines composed of. So why two lines? Because microtubules, as you know, these are hollow in nature, right? And because of that, when you project them into dimension, you essentially see them as two lines. And if you determine the distance between the two lines, that is essentially the diameter of the microtubule. So here you are able to really find out the exact diameter. And as you can see, it is a 38 nanometer, right? And 38 nanometer is combining the primary and secondary anti. Now, if you add them together, you will see that exact diameter is close to 20 nanometer, right? And that is essentially the one which is the correct diameter which people have measured by using electron microscope, right? So now you see that you are able to reach a resolution where you are able to predict the dimension of the molecules which are close to its original inherent dimension and which was impossible to see and visualize by utilizing any other methods of determining, right? You can also do this thing not only 2D, and this is the 3D image, right? And you can get a very similar resolution in 3D also. So you can clearly see how the microtubules are distributed all over the cells. And you clearly see in Z directions also, you have improvement of resolution close to the threefold of resolution, which is tremendous when you are thinking about looking at the cells. And if you are careful enough, you will be also able to see that how the microtubules are hollow in nature. This is essentially a cross sections of this three-dimensional microtube. And you can clearly see that how the microtubule structures are, which is resembles to the original what is known from the crystallography study. And as I told that you can also look at them in the multiplexed manner, and this is what you see when you do is two different DNA strands. So here the color is not from the fluorophore. You are essentially using the same fluorophore, but you are changing the interactions between these probes together. So by using the same fluorophore, you can look at two different stuff. And now it can be expanded into many more, right? And also you can look at three-dimensional also discuss. So now the last few minutes I will just take to conclude this. So not only so far whatever I have seen is on the fixed cells are not black. But if you want to go really towards more close and look at the dynamic interactions, what we did is that we use these molecules, which are, as I told, synthetic in nature and stable. When it comes to DNA, we face challenges to do this thing in the live cell because DNA are not stable. But these molecules, which I showed you, are small molecule based probes, they are able to be now very nicely be utilized, tracking the molecules in live cells. And what you see here is exactly the same thing what you have discussed in the single molecule imaging, but now it's the molecules are mobile. And that's what you see here. And these single spots are now moving. And that indicates how are you able to track these molecules in the cell, right? So you can see here that the strategy which I have applied to the fixed cell, and now it has been translated to live cell to look at the dynamics of the molecules, right? And you can do many of these statistical analysis and find out the mobility of this molecule cell surface. And those are very important for finding out how this mobility is related to cell functions, interactions with the other cells, signal transductions, and what are the advantages? As I told, you can clearly see that these molecules, because they are dynamically interacting, there is no possibility of reaching in these cells. And as you can clearly see, over time, we have imaged close to 30 minutes utilizing this strategy, and we don't see the decreased number of localities. So that means you are able to probe this molecule into its original resolutions using really high power laser because it's a single molecule imaging. But you are able to look at them really long time, right? And most of the cases, if you use the standard technique, within few minutes, the molecules are completely fixed. And that's the greatest advantage of Sarit. Okay, I think I'm going to just, as the time is over, I'm going to skip this slide. So we have also performed it's called that you can specifically tag molecules in specific part of the cells. So I'll just show you an example that, for example, if you are interested in looking at asymmetrically functionalizing molecules to the cells, this is a way to do it. So essentially what we do here is that we tag these molecules, but this molecules does not gets attached to the molecule without using light, right? So that's what you can see here. This is a complete cell. However, the complete cell is not labeled to the probes. However, only part of the cells is labeled. As also you can clearly see here, the cells are all around, but by using light, you can specifically put this molecule. So essentially what you have, not only you have a control over the molecule, but you have a spatio-temporal control, right? So you have a space control, time control of putting them up. So that can enhance your abilities. That how are you going to study single cells in a composition defined numbers? So this can also be done in many different color, as you discussed, that we can put cells in different places, different shapes, and we are able to label different cells with different color, right? And then you can now track how these cells interacts and how these cells. This can also be done in live cells also. With this, I think I would like to conclude this talk and like to acknowledge JNCSS for hosting me, providing me with all the labs and facilities. Also my colleagues who has been tremendously helpful for both collaborations as well as help as whatever needed. And also finally my group, because they have been doing the work, being the tremendous support to get these things done. And I think I'm presenting this work because they have done most of the part here, and also funding agencies, and also also Mandal for inviting symposium. And I'm very happy to address in this.
Yeah, Sarit, thanks a lot. Yeah, thanks a lot for your talk.
Sure, thank you.
It was nice and quite informative. I just thought that we should ask some questions if someone has something to ask or clarify. We can do that. So, anyone has any questions that you want to ask?
So I think there is a chat.
There is something that I thought I will ask.
Yes.
Yeah. So your CB7, is it toxic to cells?
No, not at all. So we have gone up to few micromolars to even close to millimolars, right? We have gone up to micromolars and we have not seen anything toxic. But literature of people have gone up to millimolar, and it has been widely used in many in vivo tests also. And so far we have not seen any effect in terms of the cell value.
Okay, Sarit, another thing is your CB7, is it a photo switchable or photo activable or photo shiftable? What kind of probe is this?
Okay, so the thing is that CB7 is none of these sort of an auto modulable system. Okay, so CB7 here is used as a way to attach your fluorophore to the target of interest, right? So that means that you essentially modulate that how your fluorophore interacts to your target, right? So here CB7 just acts and modulate this kind of an interactions. For example, if you are attaching fluorophore to your antibody, you will be making a covalent, right? So you will be making an amide bond essentially to conjugate it within the lysine. But here what we do, we don't conjugate it through the covalent bond, but rather we conjugate it through this non-covalent. So essentially, that is what essentially used here, not it as you act as a photomodulatable system. Is that clear?
Yeah. So I will just ask if anyone has any question, they can just pop up online. Arvind, do you see anyone online?
Yeah, there is a question like how much linkage error do we get based on these molecular tags?
Yeah, so that's a very great question. So the challenge in terms of labeling the molecules. So one area what I have not discussed is that how do you target a fluorophores or even here CD7 or anything to the specific molecular weights. And there are various ways. And one of the most popular is essentially using the antibody. And that has been useful because there are very large number of antibodies available. Essentially for every target you can get antibody, and they are quite specific and they are widely used. And however, they give linkage error as pointed out. And we are using for general purpose wherever needed that we really need to look at the molecule. We are using antibody and attaching them using the series. Right, so that gives you kind of a linkage error, what has been pointed out. However, if you think about utilizing a small molecular probe, for example, I have shown you taxol derivatives of this, or for example, a colloidal derivative of this, right? Any small molecule based probe, if you attach, I think the linkage error is going to be much more less as compared to what you will be getting using the antibody. So you will be able to localize or able to get the molecules in specifically right place as compared to using an antibody which is 10 nanometers constant. So that is, I think, advantages using the small molecule probes, and specifically CD7 and its complex are quite small. I think they just a few nanometers in diameter. So that is not going to be a problematic when it comes to the kind of linkage error.
Yeah, Sarit, we have another question. I just give.
Hello. So I have another question that you have checked the toxicity in the cell. Also, did you do any animal studies to check the toxicity?
So I have not done animal studies, but in literature report, people have injected quite high dose of CD7, and it has been shown no toxicity. So that literature is report present, but we have not done. So one of the area people use this CD7 is to essentially kind of way to extract out different other molecules. For example, many drug molecule, if there's an overdose of drug, many times people use this CD7 to eliminate the drugs because this has abilities to bind with the hydrophobic parts of the drug. So in that respect, people have used it, but I have not done. But literature report shows that it has no toxicity in animal model.
Sarit, we have one more question. That's online. How much linkage error do we get based on these molecular tags?
Yeah, so that I think I already addressed.
You already answered?
I also
Okay.
view on this.
Fine. Right. If anyone has any more questions, then we can ask.
Sir, I have a question.
Yeah.
You were talking about live cell we can image. Like, can you say some time period, 1 hour like that?
So, so the good thing about these probes are that these are dynamic images, right? That means you are not limited by the photo bleaching of the fluoride, because once a probe which is attached to a target is bleached, that can always rejuvenate with a new probe because the interactions are dynamic images, right? So there is essentially no time limit that you can do this bleach. Understand? Because as long as your fluorophore present in the solution, you can do it. Once your fluorophore is consumed, you can put a fresh load of fluorophores and it will again start doing imaging. So what we have seen is that it is really not at all based on any fluorescent bleaching property, but it is limited by what you really want. But of course, if you are using it along with really high intensity laser illuminations, that might show some toxicity, not through any of them. So yeah, I
Okay,
cannot
I guess
tell you the
there's no
exact
more
time.
question. I can just say that it is a great talk and talks a lot about the chemistry behind fluorophores, or all kinds of. Let us thank Sarit. You know,
Sure.
yeah, Sarit, thanks a lot, and we all
Thank you,
enjoyed
Bill.
this talk, and many more people are going to enjoy it online. It's also YouTube live, so I think around the globe people are looking at it,
Sure, sure.
and it will be
That's
there in the repository.
okay.
Thanks a lot. Yeah.
Thank you, sir.